Good evening, and welcome to the Science Dreams channel. Tonight's journey drifts through hibernation, a story about the moment life almost stops, but never dies. Before we begin, I'd love to know where you're watching from. It always amazes me to see listeners scattered across every corner of the planet, connected by curiosity and calm. And if you find this video soothing or interesting, please consider giving it a like and subscribing. If you're already part of this quiet little community, thank you. In several northern laboratories, researchers have watched a strange pattern repeat itself. When temperature drops and daylight shortens, the chemistry of entire organisms begins to rewrite itself in perfect sequence. Heart rate slows, oxygen use falls, metabolism folds down layer by layer, as if a switch had been thrown. Then months later, everything reverses precisely cleanly, without damage. How does a system built to run non-stop learn to pause safely? What signal tells millions of cells to conserve energy, yet keep memory intact? Why do such pauses appear in frogs, bears, insects, and even in the genes of non-hibernating mammals like us? And could this same mechanism, if fully understood, one day protect a patient after trauma, or preserve a human crew on a journey lasting years? Hibernation is not sleep, not death, not suspension. It's control. Temperature, enzymes, and hormones form a network that lowers the cost of being alive to almost zero. The blueprint already exists in nature. Science is only beginning to map its switches. Winter begins long before snow appears. It begins when daylight shortens just enough for biology to notice. Each evening arrives a little earlier. Each dawn lingers a little later. The body tracks that change with remarkable accuracy. A small timekeeping center in the brain measures the length of night, compares it to internal expectations, and sends a clear message through hormones and nerves: reduce expenditure, prepare for scarcity, and shift into conservation mode. This is the first sentence in the language of stillness. That language translates light into chemistry. During longer nights, the brain releases a nightly signal for a longer stretch, telling organs that the external world is entering a slow season. Appetite adjusts. Fuel is stored with more care. The type of fat under the skin and around organs changes in composition and purpose. Part of it becomes a special heat-making fabric within the body, designed not to move muscles, but to generate clean warmth for safe awakening later. The liver alters how it packages sugar, adding a margin for long gaps between meals. The heart prepares to beat fewer times, without losing the force required to keep tissues supplied. None of these steps are dramatic. All of them are coordinated. The rule that governs everything is simple: minimize cost, preserve structure. Chemical reactions that power cells slow with temperature. In basic biochemistry, many reactions proceed far more slowly after only a modest cooling, which means the same reserve of fuel lasts longer. Hibernation makes disciplined use of this principle. Body temperature drifts downward within a carefully chosen range. The brain does not relinquish control. It lowers set points across the network, asking every organ to idle rather than to race. This is not sleep. It is a scheduled reduction in demand that keeps essential functions intact while letting non-essential work pause. Coordination is the central problem, and nature solves it with layered timing. First comes the seasonal signal from light. Then comes a cascade of hormones that alter taste for food, how much heat the body makes, how quickly the stomach empties, and how sensitive tissues are to sugar in the blood. The pattern reshapes the whole economy of the organism. Heart muscle reduces its oxygen needs by spacing contractions farther apart, while increasing the volume of blood moved with each beat. Vessels relax, differently distributing flow where it matters most. Blood chemistry shifts toward a slightly thicker fluid that still moves smoothly at low temperature, with regulators in place to keep clotting from misfiring. Oxygen-carrying proteins alter their grip to release oxygen efficiently, even when breaths are rare. These are small changes, but together they create a safe corridor through cold. Inside cells' membranes face a special challenge. Chill can make them brittle. The solution is to alter the mixture of fats so that the surface remains flexible at low temperature. Mitochondria, the power stations of cells, shift how they handle fuel to reduce the stray byproducts that cause wear. Molecular caretakers patrol proteins to keep them folded properly while everything runs slower. Waste-clearing systems continue at a quiet pace, sweeping up what would accumulate if activity simply halted. The point is not to do nothing. The point is to do only what must be done to maintain integrity until conditions improve. The brain orchestrates the slowdown, but never goes silent. Electrical activity settles into large, slow rhythms that use little energy yet maintain timing across regions. Sensory systems dim. Circuits responsible for immediate action step aside. Memory traces are not overwritten. They are stored with care while synapses downscale to an energy level that can be sustained for a long interval. When warmth returns, those traces are ready to amplify back to daytime strength. This resilience, dim without erasing, may be the most striking feature of the state. The immune system follows its own schedule. Constant vigilance consumes fuel and can damage tissues if it continues at full force when food is scarce and wounds are rare. In the low-power season, active defenders withdraw from circulation and rest in sheltered tissues. Inflammation quiets. The system does not forget what it has learned. It simply pauses expression. When the season turns, immune cells reappear in order without a chaotic surge. The restart looks less like a spontaneous alarm and more like a neat reboot. Meanwhile, the digestive tract faces months with little or no incoming food. The community of microbes that ordinarily help to break down meals also changes. Species that live on a steady diet of plant matter fade. Others that can survive on internal secretions become more common. They produce compounds that protect the gut lining and discourage invaders, rather than specializing in extracting calories. When feeding resumes, the community flips back quickly. It is a seasonal partnership. During quiet months, protect. During active months, digest. Entry into deep slowing never happens in one step. The sequence follows a staircase. First, the animal reduces activity and shortens foraging. Nights lengthen. Small drops in temperature become tolerated, then preferred. Metabolism trims unnecessary pathways. Only after these preparations does the major descent occur. The descent itself is measured: a gentle drop in core temperature, a measured fall in heart rate, a spacing of breaths, each constrained by safety limits set by the brain. If external weather becomes unexpectedly mild, the descent pauses or reverses. If a sudden cold front arrives, the descent deepens. Flexibility prevents overcommitting to one trajectory. Return to activity is equally scripted. Warmth from the body's internal furnace activates first around the heart and brain. Blood vessels open in a mapped order so that the first rush of flow reaches critical areas without overwhelming them. The heart's rhythm tightens. Breathing becomes regular. Only then do limbs regain full tone, and voluntary motion and feeding behavior returns. The entire ascent avoids shock by restarting systems in the same order they were dialed down. How does the body know when to begin that ascent? Light provides the long message. Local cues provide the short ones. The smell of thawed soil, tiny vibrations from melting water, and the chemistry of surrounding air all change as winter fades. Those signals travel through nerves and blood toward the master timekeeper, which integrates them with the seasonal calendar before sending the order to rise. Precision matters. Waking too early risks scarce food and wasted reserves. Waking too late surrenders the first wave of opportunity. The best timing lies between, and the internal system seeks that point year after year. This entire strategy evolved because it saves energy without sacrificing structure. A modest reduction in temperature produces a large reduction in cost. Reducing speed prevents wear. Allowing long intervals between maintenance tasks gives repair enzymes time to catch up. Measurements after the quiet season often show cleaner proteins, fewer chemical scars, and rebalanced inflammation. In effect, the pause is also a reset. That reset does not require new machinery. It requires timing. Human physiology still responds to the same seasonal cues, just less dramatically. Many people notice longer sleep in darker months, slightly lower body temperature at night, and small changes in mood or appetite. Those shifts are distant echoes of a deeper program that our lineage does not fully use. The machinery that reads the sky and coordinates organs remains in place. Medicine studies it because the logic is useful. If clinicians could trigger a controlled reduction in demand for a short period, they might protect the brain after loss of circulation, preserve organs longer outside the body, or carry a patient safely through lengthy procedures. The target is not a frozen halt. It is a calm, reversible slowdown with the same safeguards nature already employs. 2 questions guide current research. How low can temperature go while keeping membranes flexible and proteins stable? And how synchronized do organs must be to avoid errors when restarting? The answers point to membranes with adjustable composition, to power stations tuned to make fewer damaging byproducts, and to circulation patterns that deliver oxygen precisely where it is needed during sparse breathing. They also point to a strict order of operations on the way back up. The system behaves like a well-written checklist. Dim this, then that. Warm this, then that. Confirm. Stability. Proceed. Snow lies quiet across the northern forest, sealing every hollow and stream beneath a surface that glows blue in the half-light. The air holds no sound except the faint creak of frozen bark. Beneath this silence, a small creature rests in a shallow pocket of earth. Its body is the color of the leaves it once hid among. Its eyes are closed, and its skin is cold to the touch. If one were to hold it, it would seem like a stone that used to breathe. Yet inside this stillness, life has not ended. It waits. This frog, no larger than a thumb, practices one of nature's most improbable equations. When frost reaches the soil, water begins to form crystals around it. Inside most living bodies, that process would tear cells apart as sharp ice pushes through membranes and concentrates salts until the chemistry of life collapses. The wood frog has found another path. As the temperature falls, its liver floods the bloodstream with simple sugars. The concentration of glucose rises hundreds of times above normal enough to pull water out of the cells and into the spaces between them. There, the first ice forms outside, not within. Inside each cell, the sugar binds to proteins and membranes, locking them into a flexible glass rather than a brittle crystal. The heartbeat slows, the blood stops moving, and within hours, the frog becomes hard to the touch. Half its body water is ice, yet every cell remains intact. This is not a random reaction. It is a sequence rehearsed with every autumn. When the nights lengthen, signals move from the brain to the liver, adjusting enzymes so that stored glycogen can be released in a rush of glucose. Small molecules like urea, which normally leave the body as waste, accumulate instead and add a second layer of protection. Urea stabilizes proteins and prevents excessive water loss during freezing. As the temperature drops below zero, these two solutes, glucose and urea, create a biochemical shield that keeps water where it is safe. The result is an organism that can tolerate conditions that would end almost any other vertebrate's metabolism. When the frog is frozen, its heart no longer beats. Blood no longer circulates. Oxygen delivery ceases. The cells inside shift into a silent mode, relying on what little fuel remains and tolerating the absence of oxygen for days, even weeks. Metabolism slows until only fragments of activity persist. Enzymes fold their arms, membranes stiffen slightly, but do not burst, and electrical gradients across cell walls fall to near zero. The organism has reached the edge of reversible death, and yet it does not cross it. In spring, warmth arrives first through sunlight. The upper crust of snow softens water, begins to flow between the layers of ice, and the soil breathes again. The frog senses this through temperature-sensitive nerves that remain functional even near freezing. The first sign of revival is subtle, a small pulse of heat as enzymatic reactions restart in the liver, turning stored glycogen back into glucose. The ice begins to melt outward from the heart, then the abdomen, then the limbs. Within an hour, blood moves again. The heart starts to beat slow at first, then steady. The frog takes its first breath of the new season and resumes the work that winter had paused. What seems miraculous is chemistry organized in time. The limits are strict. Ice must form slowly enough to allow water redistribution, and melting must follow the same discipline. A temperature drop too steep can trap water inside cells, forming fatal crystals. A rise too fast can cause rehydration, shock swelling membranes before they regain flexibility. The wood frog survives because its entire body behaves as one coordinated system, a collaboration between tissue types that never forget their roles even while inactive. Researchers studying these animals have found direct parallels to human medicine. When surgeons cool organs for transport, they face the same problems of ice formation, dehydration, and salt imbalance. Cryoprotectants derived from the frog's chemistry, simple sugars, nitrogen compounds, and antifreeze proteins from other cold-adapted species, now inform how human organs are preserved between donors and recipients. Scientists explore whether small controlled amounts of these molecules might protect cells during oxygen loss after a heart attack or stroke. The idea is not to freeze people, but to borrow nature's precision in slowing time safely. Ice, for most of life, represents an absolute boundary. The frog treats it as a phase to manage, not an enemy to resist. It neither hides from freezing nor attempts to stay warm against all odds. It reorganizes itself so that freezing becomes a part of living. This is not courage or adaptation in the emotional sense. It is geometry, chemistry, and timing brought into alignment through generations. The same principle appears at smaller scales. Inside the cells of the frog, specialized proteins attach to the faces of forming ice, halting their growth. They create a narrow window of stability where ice can exist without spreading. The frog's own body fluids exploit this gap, existing partly frozen, partly liquid, a state between phases. Water becomes structure without becoming hazard. For months, that structure holds. When the thaw comes, the proteins release their grip, and the transition back to liquid proceeds without damage. In recent years, scientists have begun to use the term controlled vitrification to describe similar transitions in the laboratory. It means creating a glass-like solid without ice suspending molecules in place without destroying them. This principle has already allowed certain cells and embryos to be preserved for long periods. The challenge remains scale. A frog can achieve it across its entire body because its tissues are small, its vascular system simple, and its natural cryoprotectants evenly distributed. Applying that to larger organisms requires precision still beyond current engineering. But the physics is the same. In the quiet underlayer of snow, such complexity happens in absolute stillness. The frog does not twitch or tremble. It waits, and waiting itself becomes the act of survival. To the eye, it appears gone. To chemistry, it remains in negotiation. Every molecule holds its position until the sky returns to blue and the water around it flows again. Then, as if rewinding a film, life resumes exactly where it left off. The forest deepens as winter reaches its middle. Light lingers low, casting long blue shadows across the snow. Beneath a slope lined with spruce and birch lies a hollow in the ground, its entrance hidden beneath a curtain of ice and moss. Inside, the air is still and cold, yet not freezing. A slow warmth radiates from the creature curled within. The earth around it is pressed smooth by its weight. The surface fur glistens faintly with condensation that freezes and melts again in slow cycles. The bear is motionless, its great chest rising only a few times each minute. From outside the den, it could be mistaken for stone. Inside, its body conducts a symphony of silence, more intricate than any movement of wakefulness. The bear's winter state is not a sleep, but a reorganization. In the months before snow, it eats constantly, turning berries, roots, and small prey into dense stores of fat. As daylight shortens, the internal chemistry shifts. Hormones that control appetite fade. Enzymes that regulate energy switch direction. The liver begins to build long chains of stored fuel, while other tissues prepare to rely on them. When the bear finally retreats underground, it carries enough energy to sustain it for more than half the year without a single meal. Then begins the gradual descent. The heart, which might beat 60 times each minute in summer, falls to a fraction of that pace. Breathing slows until many minutes pass between inhalations. Body temperature declines by several degrees, not enough to freeze, but enough to slow nearly every reaction. Yet the bear remains warm compared with smaller hibernators. It occupies an intermediate zone where energy is saved, but the systems of the body never truly shut down. The challenge is not surviving cold, it is surviving stillness. For most animals, months without movement would bring decay. Muscles waste away when unused bones lose calcium and circulation falters. In the bear, none of this occurs. The body has evolved to recycle itself in real time. Urea, the nitrogen waste that in other species is excreted, is instead reabsorbed and broken down. The nitrogen from it feeds the synthesis of new amino acids, which in turn maintain muscle fibers and internal proteins. The bear lives on its own molecular leftovers, closing a loop between breakdown and renewal. Scientists studying this process have found that the balance between protein destruction and creation stays remarkably stable throughout hibernation. When the bear stands in spring, its strength is nearly unchanged. The same conservation extends to bone. In human bed rest, even a few weeks of immobility can lead to measurable bone loss. In the bear's winter months pass with no weight bearing, yet bone density remains intact. The explanation lies in signaling molecules that modulate the balance between bone forming and bone-resorbing cells. During hibernation, both sets of cells reduce activity in equal measure, preserving structure without waste. Calcium and phosphate circulate in closed cycles, never drifting far from equilibrium. When activity resumes, the skeleton awakens, ready to carry the same weight as before. This preservation comes with a quiet choreography of blood and heart. The heartbeat is slow, but each contraction is powerful, pushing nearly the same amount of blood as in summer. Blood itself becomes slightly more concentrated, allowing oxygen to reach tissues efficiently even when flow is reduced. The chemistry of clotting adjusts as well, lowering the risk of stagnation in slow-moving vessels. Red blood cells linger longer, yet their membranes remain supple. The result is a circulation system tuned for stillness, steady, economical, precise. The immune system rests too, but not idly. Its vigilance drops, reducing inflammation and saving energy, yet its memory cells remain ready to act. The body lowers its defensive tone without abandoning recognition. When the bear finally reawakens, the immune response rises in ordered steps, avoiding the storms of inflammation that might follow months of inactivity. Even the gut's microbial residents participate. As food intake halts, the community reorganizes, favoring species that can survive on mucus and cellular debris rather than fresh plant matter. These microbes, far from dormant, produce short-chain fatty acids that nourish the cells lining the intestine and help maintain barrier integrity. The digestive system sleeps, but the fabric of it does not unravel. Within the den, the air is thick with humidity and the faint scent of soil. The bear's body temperature hovers near the edge, where chemical reactions are still reliable. Every few weeks it shifts, slightly rolling to ease the pressure on one side, then returns to stillness. Its cubs, born in midwinter, while the mother remains half asleep, feed and grow in warmth created entirely by her metabolism. Their hearts race at a summer rhythm, while hers continues its winter pace. Two tempos nested within one body keep time together until spring, when the snow outside softens and daylight lengthens. The first signal to awaken arrives. The brain senses the change through subtle variations in light, filtering through closed eyelids, and through fluctuations in hormone patterns tied to day length. Metabolism stirs. Heat begins to rise from specialized stores of brown fat, clustered near the shoulders and spine. This tissue, rich in mitochondria, releases energy as heat, rather than as chemical work. It ignites the thaw from within. The first warmth flows to the heart and brain, restoring coordination before the limbs move. Circulation quickens, respiration deepens, and by the time the outer snow begins to melt, the den has become a chamber of awakening life. The bear steps out not as something reborn, but as something continuous. Months of stillness have passed without erosion. The systems of its body have balanced use and repair so exactly that no debt of recovery remains. What looks like sleep was maintenance performed at the lowest possible cost. For scientists, this equilibrium offers a model. If human metabolism could be adjusted to mimic these processes, patients immobilized after injury might avoid muscle loss. Astronauts could travel long distances without bone decay. Even organs awaiting transplant might survive longer if stored in a state that preserves structure while reducing energy demand. The key lies not in deep freezing or radical transformation, but in controlled quiet, the same principle that lets a bear endure half a year inside a sealed chamber of earth. Outside the den, the forest begins to echo again. Meltwater trickles down the slope, birds return to the canopy, and the air smells of thawed resin. The bear lifts its head toward the light, blinking against brightness. Its movement is slow but deliberate. Muscles that have not flexed in months respond as though they never stopped. The long silence ends, replaced by the faint rhythm of paw steps on wet ground. Life, having mastered stillness, resumes its steady motion. In a limestone cave above a frozen river, winter breathes through narrow cracks in the rock. The air inside is cold enough to numb skin, but the temperature barely shifts from night to day. Walls glisten with thin ice, and drops of water fall in slow intervals, each echo carrying through the hollow like a clock that measures silence instead of time. From the ceiling hang thousands of small bodies, each wrapped in its own skin like a folded leaf. They sway gently in unseen currents, their claws clinging to the stone. To the eye, the colony seems lifeless, a dark pattern carved from the rock itself. Yet within every folded shape, there is a heartbeat, so slow, it can almost be mistaken for none at all. Bats enter this state together, but not by chance. Each autumn they gather in the hundreds or thousands, drawn to caves, tunnels, and abandoned mines where the air remains steady. They form dense clusters, conserving both space and heat. As outside temperatures fall, their metabolism begins to decline in unison. Heart rates that once beat hundreds of times each minute fall to single digits. Breathing slows until minutes pass between breaths. Body temperature drifts downward until it is barely above the air around them. Within this narrow range, energy consumption drops by nearly 100-fold. The colony becomes a quiet field of suspended engines, each burning just enough to keep chemistry reversible. The bat's survival depends on precision. Every gram of fat stored during autumn determines how many nights they can last until spring. To reduce consumption further, they maintain an extremely low metabolic rate for long intervals, interrupted only by brief awakenings. These awakenings are costly yet necessary. During them, the bat's temperature rises quickly to its summer value. Its heart resumes a racing rhythm, and its body repairs what months of torpor have neglected. Enzymes that clear metabolic waste resume their work. Immune cells patrol. The brain resets its balance of ions and transmitters. After a few hours, the system cools again, returning to near-frozen stillness. It is a rhythm written across weeks-long spans of silence, punctuated by short, vital pulses of life. Inside the muscle of the heart, chemistry adjusts to make this possible. The cells change the ratio of proteins that handle calcium, ensuring contractions remain efficient even at near-freezing temperatures. Ion channels across the cell membranes slow their gating to match the cold, preventing electrical instability. Mitochondria, the tiny engines within each cell, shift fuel preference from carbohydrates to fats, producing more energy per molecule at a slower pace. The heart becomes a low-speed, high-efficiency machine that can pause almost completely and still resume without injury. The communal aspect of bat hibernation adds another layer of complexity. Each individual's warmth influences the others. The clustered group creates a microclimate where temperature stays stable and humidity remains high enough to prevent desiccation. Air circulation within the cave, guided by narrow entrances, and the density of bodies keeps oxygen from falling too low. Together, the colony forms a single breathing organism whose temperature fluctuates by only a few degrees all winter. The structure is self-regulating. If one section becomes too cold, small movements ripple through the cluster, redistributing warmth without waking the group. What appears static is a delicate balance of shared heat and shared restraint. The environment itself must also cooperate. If temperatures rise too early, or if humans enter the cave, even brief disturbances can trigger premature awakenings. Each burst of activity consumes a significant fraction of the stored fat, leaving less for the remaining months. A few such interruptions can make the difference between survival and starvation before spring. Across continents, researchers have documented how subtle changes in cave microclimate or the presence of disease can push colonies toward collapse. The fragility of this quiet economy is matched only by its precision. The heart of a hibernating bat offers clues about resilience under near stasis. During torpor, its cells tolerate conditions that would stop a human heart within minutes. Low oxygen, slow blood flow, and long pauses between contractions. The key lies in how its enzymes and structural proteins resist the cascade of damage that such stress would cause in other mammals. Instead of producing harmful oxidants, the bat's mitochondria remain tightly coupled, leaking almost no reactive molecules. The heart's internal scaffolding retains flexibility, preventing the stiffness that would otherwise accompany prolonged cold. When the animal warm circulation restarts smoothly, without clots or inflammation, the transition between torpor and wakefulness is so clean that the heartbeat itself leaves no trace of the transformation. Every few weeks, a faint stirring passes through the colony. One or two individuals begin to vibrate, generating heat through rapid muscle contractions. Their temperature rises, triggering others nearby to stir. In a matter of minutes, entire clusters flicker awake, each heart accelerating from a few beats per minute to hundreds. The air fills with a soft rustle, then quiets again as they settle back into stillness. These cycles are synchronized across the winter, perhaps guided by internal clocks, still sensitive to the length of darkness outside. The pattern ensures that each animal's systems receive maintenance without wasting the reserves that must last until the first insects return. Scientists studying these rhythms have begun to draw parallels with artificial hypothermia used in human medicine. When surgeons cool patients during heart or brain procedures, they rely on similar reductions in metabolic rate to protect tissue from oxygen loss. Understanding how bats restart their hearts without damage could lead to safer rewarming methods and improved recovery after cardiac arrest. The same principles might one day inform long-term space travel, where metabolic suppression could conserve resources for journeys measured in years. Nature has already written the blueprint. What remains is to learn its syntax. In the cave, the long winter drifts toward its end. Water begins to trickle along the floor, carrying thin layers of sediment from the ceiling. The air grows slightly warmer, though still cold enough to sting. The bats remain asleep, each suspended in its private silence. Then, on a day that seems no different from the last, the first one opens its wings. Muscles stretch, the body shivers once, and the heart accelerates to its full rhythm. Others follow, unfolding from the ceiling like petals in reverse. The sound builds from scattered rustles to a low, continuous hum. When they finally lift into the air, the motion is smooth, unhurried, as if continuity had never been broken. Outside, the world is wet and pale, the snow soft under the first true sunlight. The bats spiral upward, feeding on early insects rising from the thaw. The heart, that once slowed to the edge of stillness, now beats freely again, carrying the rhythm of a season's worth of patience. As night returns, the cave empties. Silence settles once more, leaving only the faint drip of water and the echo of wings. The season of waiting has ended, and the forest begins to stir with other forms of quiet endurance. Farther north, where the ground never fully thaws and the temperature sinks below what even a bat can tolerate, another creature takes the experiment further, balancing on the boundary between liquid and solid life. In the far north, where the sun slips below the horizon for months, winter does not simply arrive. It settles like geology. The ground remains locked in frost so deep that even sound seems slower there. Wind drifts over the tundra, carrying grains of ice that never melt, and in burrows beneath the surface, small lives continue a negotiation with the cold that borders on impossible. Among them, a ground squirrel no longer resists freezing. It becomes part of it. As the final days of summer shorten, the squirrel prepares with quiet urgency. Layers of fat grow beneath the skin, not as insulation, but as a future source of steady, slow fuel. Its body calibrates a precise chemical balance, lowering concentrations of certain ions and altering the composition of cell membranes. The brain reduces its need for oxygen by silencing large regions of neural activity, leaving only circuits essential for coordination. When the first permanent frost seeps through the soil, the animal curls in its narrow chamber and begins its descent into one of biology's coldest states. The internal temperature drops degree by degree until it passes the freezing point of water. Unlike the frog, the squirrel's body fluids do not crystallize. Instead, they reach a supercooled state liquid, below zero, stabilized by specialized proteins and sugars that prevent ice nucleation. Every heartbeat grows faint, every breath slower, until hours can pass without either. Within the chest, the heart shrinks slightly and moves only enough to preserve integrity. The rest is stillness, but not death. At this threshold, survival depends on control. The animal's cells contain a mixture of small molecules that act as cryoprotectants, substances that bind water, reducing its ability to form crystals. These include glycerol glucose and a family of antifreeze proteins that attach themselves to any microscopic ice seeds that might appear, halting their growth. The result is a metastable state, fragile yet durable, in which the body remains unfrozen, despite ambient temperatures far below zero. In this suspended condition, metabolism falls to a fraction of normal activity. Energy consumption nearly vanishes. The organism becomes a quiet equation of chemistry and time. While external cold deepens, internal order endures. Mitochondria shift their behavior, minimizing leakage of reactive molecules that would otherwise damage tissue. The immune system withdraws but keeps its structural memory intact. Even DNA repair enzymes remain faintly active, patching errors that occur during months of quiescence. The heart, which might have failed under such stress in any other mammal, retains its delicate architecture by adjusting calcium handling and by keeping cell membranes soft. Each mechanism serves the same goal: to hold the line between life and the physics of freezing. Occasionally, the squirrel's body must return to higher temperature. Every few weeks, a slow stirring begins from the core. Heat rises not from movement, but from the controlled ignition of fat stores in brown adipose tissue. Oxygen use increases, circulation resumes, and the animal's temperature climbs briefly to near normal. During these short intervals, lasting less than a day, accumulated waste is cleared, electrolytes are balanced, and the brain performs its own quiet maintenance. Then, the cycle reverses. The animal cools once more, its chemistry folding back into stillness. This oscillation between near death and wakefulness continues throughout the winter, each pulse timed by an internal clock that measures months of darkness. What the squirrel achieves pushes the limits of physiology. In laboratory measurements, core body temperatures of some individuals have fallen below -2 degrees Celsius. Yet tissues remain intact. The boundaries of biochemistry bend but do not break. Proteins that would denature under such stress remain stable because their surroundings have been engineered at the molecular level to protect them. Water behaves differently in the presence of these solutes, slower, thicker, unable to assemble into lattices of ice. Inside this altered medium, life persists in a kind of slow motion. Such control has implications far beyond the Arctic. Researchers study these squirrels to understand how cells endure extremes of temperature and oxygen deprivation. The answers inform experiments in organ preservation, hypothermic surgery, and even the prospect of long-duration spaceflight. If the conditions that govern this natural torpor could be reproduced safely in humans, it might one day be possible to pause metabolism without harm, extending the window between injury and recovery, or between departure and destination. The challenge lies in scale. What works for a creature weighing less than a kilogram must be translated to bodies millions of times larger without losing coherence. Back beneath the snow, the animal's world remains hushed. Soil particles gleam faintly with frost, and air trapped in ice pockets moves sluggishly through the tunnels. Each layer of insulation holds a different temperature, from just below freezing near the surface to several degrees colder in the deepest levels. Within this gradient, the squirrel positions itself, carefully adjusting its posture to regulate heat loss. Its heartbeat may pause for minutes, yet blood continues to circulate slowly through major vessels carrying enough oxygen to prevent stagnation. Even in stillness, geometry matters. The shape of coiled limbs, the curve of the spine, the orientation of the head, all serve to maintain equilibrium through the long season of darkness. When spring light finally returns, it filters through snow that has begun to thin. The warmth seeps downward until it touches the first chambers of the burrows. Their sensors within the brain detect the change and send signals that awaken metabolism. Fat combustion accelerates, muscle tone returns, and the heart's rhythm quickens. Ice melts in the surrounding soil, carrying dissolved gases that feed the returning roots of tundra plants. The animal stirs for the first time in months, emerging into a landscape that seems both unchanged and entirely new. From a scientific view, the Arctic ground squirrel's adaptation stands as the most extreme example of mammalian hibernation known. Its ability to allow core body temperature to drop below freezing without tissue injury challenges assumptions about the limits of warm-blooded life. It is a demonstration of balance between physics and physiology, a meeting point of energy conservation and structural preservation. Unlike migration, which escapes winter, or dormancy, which hides from it, this strategy uses the cold itself as a tool. The organism becomes an engineer of phase transitions, turning its own fluids into controlled environments where ice and liquid coexist under supervision. At the same time, its existence links ecosystems across seasons. Predators depend on the squirrel's re-emergence to mark the beginning of the feeding cycle. The burrows it excavates aerate permafrost soils and influence the distribution of thaw water. Even its long sleep affects the chemistry of the tundra, as stored carbon and nitrogen shift forms between active and dormant months. In this way, hibernation becomes not only a strategy for survival, but also a regulator of landscapes. The pause of one small mammal contributes to the rhythm of entire biomes. By the end of spring, the burrows empty. The snow above collapses into shallow melt streams, and the air carries a scent of wet lichen and mineral dust. The squirrels scatter across the plain, their hearts beating again at summer, speed their bodies almost unchanged despite half a year of near-frozen stasis. To an observer, the transition appears effortless, as if time had folded over itself. In truth, it is the result of unbroken discipline, an economy of energy measured to the last molecule. In the larger arc of evolution, such control marks one of life's rarest achievements, the ability to pause without decay. Across billions of years, organisms have found countless ways to endure scarcity. Yet only a few have learned to stop almost completely and still return. The Arctic ground squirrel lives at that threshold, carrying within it a lesson about precision. It reminds us that survival is not always motion, not always struggle. Sometimes it is the grace of doing nothing until the world itself changes enough to welcome motion again. As the tundra brightens under the long light of spring, other creatures stir in hidden waters. Beneath a film of ice where oxygen has long been absent, an older lineage waits. It neither breathes nor freezes. Its shell holds the memory of summer sun, its blood holds the chemistry of endurance, and its heart beats only a handful of times each hour. The next story lies there beneath the thawing ponds, where the art of surviving without breath begins. At the bottom of a frozen pond, light filters weakly through the snow and ice above, turning the world beneath into a dim green stillness. No ripples disturb the surface. Stems of waterweed stand motionless, trapped inside glass. The temperature hovers just above freezing, and oxygen has long been consumed by decay and the slow chemistry of mud. Yet within this breathless silence, life remains. Beneath a thin layer of silt, a painted turtle lies buried, its shell pressed against the dark sediment, its head tucked beneath one front limb. To any observer, it would seem lifeless. But inside its chest, a heart still beats, though so slowly that minutes may pass between contractions. Unlike mammals or amphibians that avoid freezing, the turtle accepts what the pond dictates. As winter closes in, the water surface locks into ice, sealing away the exchange of gases with the air. Oxygen levels in the pond fall until they approach zero. For most aquatic animals, this would mean suffocation. The turtle answers with patience. Its body switches from oxygen-based metabolism to a slower anaerobic mode, one that yields far less energy but requires none of the air that no longer exists. Every reaction that normally burns fuel with oxygen now relies on glycolysis, breaking sugars into lactic acid and tiny bursts of usable energy. It is a costly compromise. Energy output drops to a fraction of what it was, and the waste acid accumulating in the blood must be managed with precision. Here, the shell plays a second role. Usually seen as armor, it becomes chemistry's ally. Made largely of calcium carbonate, it acts as a buffer, absorbing and neutralizing the lactic acid produced during the months of anoxia. The bones of the skeleton join in releasing small amounts of minerals that bind to the acid and keep blood pH within a survivable range. The turtle's tissues, instead of drowning in acidity, glide through a balanced quiet. The shell itself changes color slightly. As the season progresses, taking on a duller hue, its stored calcium, spent in this slow trade with the internal chemistry of survival. Heart rate declines to perhaps 1 or 2 beats each minute. Blood flow concentrates around the brain and vital organs, while most muscles cool and fall into deep dormancy. Nerve cells that would otherwise demand constant oxygen reduce their firing to nearly nothing. Ion channels in their membranes close, trapping charge within and preventing the leaks that would cause exhaustion. The brain's activity pattern becomes sparse, marked only by occasional bursts that seem to test the system's readiness. There is no dream, no awareness, only maintenance. Respiration, as a mechanical act, drawing air, exchanging gases, ceases entirely. Yet the body continues to move small amounts of water and ions across its surfaces. Oxygen that diffuses through the skin, the cloacal lining, and the inner mouth reaches the blood, slowly supplementing what little remains dissolved in the pond water. This passive exchange keeps a trace of aerobic metabolism alive, even in the darkest days. The turtle's tissues have evolved to use every molecule efficiently. Hemoglobin binds oxygen, tightly releasing it only when concentration drops to almost nothing. In this marginal state, the difference between life and death is measured in single breaths that never happen. Temperature is both danger and protection. As the body cools to near the freezing point, chemical reactions slow, reducing energy demand. The viscosity of fluids increases the movement of molecules through membrane slows, and the signals that coordinate heart and muscle stretch into long intervals. The slowdown is not random. It follows an ancient schedule written in the physiology of the species. When temperature falls too low, enzyme systems reconfigure to remain active within narrow limits. When it rises again, they fold back into their warm weather forms. The same proteins work across seasons, adapting their flexibility to whatever physics demands. For months, this equilibrium holds. No sound enters the pond except the deep groan of shifting ice above. Silt settles over the turtle's shell, and small bubbles collect along its edges, traces of metabolic gas rising slowly toward the surface. Energy reserves decline, but not disastrously. The liver releases glycogen, the stored sugar that fuels anaerobic metabolism in measured pulses. Fat stores remain mostly untouched, waiting for the return of oxygen to burn efficiently. Nothing is wasted. Even the brain's temperature fluctuates slightly, cooling a few degrees below the rest of the body to further conserve energy. As spring light strengthens, cracks form in the ice above. The first meltwater trickles down through the fractures, carrying oxygen back into the pond. Sensors in the turtle's blood and brain detect the change almost instantly. Ion gradients across cell membranes begin to reset. The heart quickens by imperceptible degrees. Blood flow expands from the core toward the limbs, flushing accumulated acids into the newly oxygenated water. The shell and skeleton reclaim the minerals they lent during winter. Within a few hours, the animal's metabolism has turned back toward normal respiration. Air hunger returns, and with it movement. When the ice finally opens, the turtle rises to the surface. It breaks through the film of water with a quiet exhalation that sounds almost like relief. The first deep breath in months fills its lungs, reawakening the full rhythm of circulation. Sunlight warms its shell, drawing steam from the damp carapace. What began as stillness in darkness ends as stillness in light. The creature that seemed stone becomes motion again, not through drama, but through sequence. From a physiological view, this ability ranks among the most remarkable on Earth. Few vertebrates can survive complete anoxia for longer than minutes. The painted turtle endures it for months, orchestrating the chemistry of life at the very edge of viability. Its success relies on principles that medicine now studies, with care-controlled reduction of metabolism, tolerance of acid, build-up, protection of neural tissue during oxygen deprivation, and the use of buffering minerals to maintain balance. These same mechanisms inspire clinical approaches to organ preservation and recovery after cardiac arrest, where the aim is to slow damage until oxygen returns. Nature has already tested the methods through millennia of winters. In ecological terms, the turtle's survival also carries significance beyond itself. By remaining alive beneath the ice, it connects the summer ecosystem to the next season's renewal. When it resurfaces, it becomes both participant and signal in the pond's rebirth. Its stirring disturbs the sediment, releasing nutrients to feed the first algae bloom. Its predators, newly awakened or migrated back, depend on its return to complete their cycles of feeding and breeding. Even its waste contributes to the chemistry that restarts microbial life after months of stasis. The turtle's long, breathless sleep anchors a chain of events that radiates outward through water, soil, and air. Above the pond, thaw continues. Ice collapses in sheets, breaking into shards that drift toward the banks. The sound of running water replaces the silence of snow. On the mud, small insects rise and fall, their movements sharp against the pale reflection of clouds. The turtle rests in the shallows, eyes half closed, adjusting once again to light. Around it, the cycle of oxygen and stillness, warmth and cold begins anew. Each year repeats the same sequence, refined by evolution until it appears effortless. The lesson in this slow rhythm is simple: life's persistence is not measured by motion, but by precision. To breathe without breath is to understand limits, to find space within the narrowest margin between activity and cessation. In the turtle's calm endurance lies an ancient truth shared across all hibernators, the realization that survival is not always a contest against the environment, but a conversation with it carried out molecule by molecule until the light returns. When the frozen months finally begin to loosen their grip, warmth does not return all at once. It seeps back in slow gradients, sunlight, through clouds thawing soil from beneath the soft hiss of snow, collapsing into water. In hidden dens, burrows, and nests, the change triggers an equally quiet response. The still bodies of winter's hibernators begin to stir, not with movement, but with chemistry. Deep inside each one, heat awakens from within. For most creatures, emerging from hibernation survival depends on rewarming. Yet the act of warming is perilous. Rise too fast, and tissues rupture under sudden expansion. Too slowly, and the cold lingers long enough to exhaust dwindling reserves. Nature's solution to this delicate equation is neither muscle nor shiver. It is a special kind of tissue, a quiet furnace built for control. Within bears, squirrels, and tiny rodents alike lies brown adipose tissue, or brown fat, a dense mitochondria-rich fabric whose sole purpose is to produce heat without motion. Unlike ordinary fat, which stores energy, brown fat consumes it. Each cell is filled with mitochondria, those small double-walled organelles that transform nutrients into chemical power. In most tissues, mitochondria produce energy in the form of molecules that fuel muscles and organs. In brown fat, that process is rewired. A unique protein uncouples the flow of energy from work, allowing it to escape as heat instead. The reaction hums silently through the animal's body, converting stored fat into warmth that radiates outward like a soft current. There is no flame, no tremor, only the soundless acceleration of chemistry. The trigger for this transformation is precise. As daylight returns and temperatures rise by only a few degrees, nerves branching from the brain send signals through the sympathetic system to brown fat deposits around the neck, shoulders, and along the spine. These signals release molecules that activate the uncoupling proteins inside the mitochondria. Within minutes, temperature in those regions begins to climb. Blood vessels widen to carry heat to the heart and brain, first ensuring the core warms before the extremities. The heart, which has idled through winter, responds immediately. Each beat pushes the new warmth farther, expanding its reach through the organism's returning life. In a bear's den, the process unfolds over hours. The great body remains still while its internal temperature climbs. Muscles twitch occasionally as circulation expands, but there is no struggle. The animal's metabolism has learned the path back to wakefulness. The same hormones that once slowed its systems now reverse direction, increasing the flow of thyroid signals that raise metabolic rate. Blood chemistry adjusts, balancing electrolytes that shifted during the long rest. The first true breath of spring air will not come until the inner temperature stabilizes. Only then does the bear begin to move its eyes, opening to light that has returned by measure. For smaller hibernators, the rhythm is faster, but no less orderly. A ground squirrel may rewarm from sub-zero to full activity in a matter of hours. The brown fat burns through its limited stores in an exact sequence, lifting body temperature, enough for shivering to resume a second wave of thermogenesis that amplifies the effect. The animal's muscles join the process, converting chemical energy into kinetic heat. The two systems overlap, ensuring that reanimation proceeds evenly. When at last the body reaches its normal temperature, every cell begins again the work of metabolism, as if the intervening months had been nothing more than a long-held breath. This mastery of controlled heat has meaning beyond winter. In humans, remnants of brown fat linger most active in newborns who cannot yet shiver, and in small deposits along the spine and neck of adults. For decades, scientists assumed these cells vanished with age, but imaging studies revealed otherwise. Under cold conditions, even adult bodies can activate brown fat consuming glucose and generating warmth without movement. Its presence has reshaped our understanding of metabolism. Research now explores how stimulating these tissues could help regulate energy balance, improve insulin sensitivity, or counter obesity. The same molecular switch that wakes a hibernator might, in gentler form, refine human physiology. Rewarming after hibernation is more than heat production. It is orchestration. As circulation expands, oxygen floods into tissues that have been cold and slow for months. Too much, too quickly, can damage delicate structures through oxidative stress. To prevent this, the body times the sequence carefully. Antioxidant systems activate before full reoxygenation, neutralizing reactive molecules as metabolism accelerates. The heart resumes full rhythm only after blood chemistry stabilizes. The lungs adjust, gradually reopening alveoli that had partially collapsed during torpor. Even the brain, the hungriest organ of all, waits a few minutes before resuming its higher functions. Each part returns in turn, guided by signals older than the species themselves. Outside the den, the world echoes this reawakening. Streams release from their ice casings, running clearer and louder each day. The first insects drift in slow, uncertain paths above puddles of meltwater. Buds swell along the branches, their sap driven upward by the same rising warmth. The synchronization is so precise that one could almost mistake it for coordination, a planet-sized metabolism turning itself back on. In truth, each system answers only to physics and light, but together they form a pattern as intricate as any heartbeat. The beauty of brown fat lies in its simplicity. It does not store complexity or intelligence, only efficiency. It turns one form of energy directly into another with almost no loss. The process wastes nothing. What might seem trivial, a patch of tissue a few grams in weight, can decide the difference between life and death in the fragile hours of awakening. Without it, many hibernators would never rise from their long silence. They would remain trapped in the equilibrium of cold, unable to cross the narrow bridge back to motion. Even after full activity returns, echoes of hibernation remain. For weeks, metabolism stays slightly lower than its summer pace, conserving energy, while food remains scarce. The transition from stored reserves to fresh intake proceeds gradually. Appetite returns only after digestive bacteria themselves, dormant through winter, repopulate the gut. The body tests its systems one by one, as though remembering the rhythm of motion after too long still. Each species performs the same quiet ritual, calibrated to its environment. There are no mistakes here, only the measured reopening of life. Spring deepens until frost becomes memory. The last shards of ice vanish beneath soil and water, and in the renewed warmth, life performs its quiet maintenance. What hibernation slowed must now be restored. The systems that held still for months begin their reawakening in careful order. Among these, none are more intricate than the immune system and the unseen communities of life inside the gut. During hibernation, the body's defenses rest. The immune system, a network of organ cells and molecular messengers, consumes immense energy when active. Each patrol of white blood cells, each release of chemical signals that mark and remove invaders, demands fuel. In the long, cold months when food is absent, such vigilance would be unsustainable. The body's solution is not to abandon defense, but to suspend it, entering a phase of near silence. White blood cells retreat from circulation into reservoirs within bone marrow and lymph nodes. Production of new cells slows, inflammation fades, and molecular alarms quiet. The internal landscape becomes calm, still alive, but subdued. In this state of dormancy, the risk of infection might seem high. Yet, most hibernators spend winter sealed in isolation, beneath snow, inside dens, or underwater, where exposure to pathogens is minimal. The body's lowered temperature further limits microbial activity. Viruses and bacteria that might thrive in warmth find no foothold in the cold. In this way, stillness itself becomes protection. Energy that would have fueled defense is saved for the long endurance of fasting. When the body begins to wake, the immune system must do the same. The return is delicate. As circulation quickens and temperature rises, dormant cells sense new chemical cues. Cytokines, small signaling molecules, spread through the blood like distant echoes returning to the surface. Stored white blood cells re-enter the bloodstream, ready to resume their patrol. In bears and ground squirrels, studies show that within hours of rewarming, immune markers return to active levels. The transition is so smooth that it seems less a restart than a continuation delayed. The pattern resembles a computer that reboots after long sleep. Circuits re-engage, checks are performed, and memory is verified before new tasks begin. The immune system, too, verifies its integrity. Cells that might have aged or accumulated damage are quietly removed through programmed death, replaced by new ones from reserves. Organs, such as the spleen and thymus, expand slightly as production resumes. Within days, the system that had almost vanished during hibernation hums again, tuned to a body, now ready for exposure to the living world. Yet the immune system does not act alone. Inside every animal, trillions of microbes inhabit the gut, a second community whose fate mirrors that of its host. During months without feeding, their environment changes completely. Food-derived nutrients vanish, oxygen levels shift, and the slow peristaltic rhythm of digestion halts. Many bacterial species die back. Others take their place, feeding on mucus and sloughed intestinal cells instead of plant or animal matter. The gut becomes an ecosystem of specialists in scarcity. This transformation, though severe, is not random. In ground squirrels, for instance, microbial communities reorganize into a composition that favors energy extraction from internal sources and suppresses inflammation. Certain groups of bacteria even produce short-chain fatty acids that nourish the cells lining the gut wall, keeping the barrier intact. By reducing the population of microbes that would otherwise trigger immune responses, the dormant gut stays protected. The body, the immune system, and its microscopic residents form an alliance based on stillness. When spring returns and feeding resumes, a second wave of transformation occurs. New food enters the digestive tract for the first time in months. With it come fibers, sugars, and proteins that dormant species cannot process. Dormant spores awaken and bacterial diversity expands again. Within days, the microbial landscape shifts back toward its summer configuration. The process resembles ecological succession. After a forest fire, first the hardy colonizers, then the full community, each stage laying groundwork for the next. The gut reclaims its function, and in doing so, signals the immune system to resume its calibration. The two systems communicate continuously, balancing tolerance and vigilance. From an evolutionary perspective, this cycle of shutdown and renewal offers powerful advantages. It conserves resources during famine and reduces the risk of autoimmune damage, a common consequence of an immune system that never rests. By temporarily silencing inflammatory pathways, hibernators avoid chronic stress on their tissues. Researchers studying bears have found that even after months of fasting, their muscles show minimal signs of inflammation or degradation. The same molecules that suppress immune activity in winter may also protect against age-related diseases. The body uses hibernation not merely to survive cold, but to clean itself of the wear accumulated through motion and metabolism. In human medicine, these insights have begun to echo. Understanding how animals pause and restart immune function could inspire therapies for autoimmune disorders, where the body mistakes its own tissues for threats. Scientists are exploring whether inducing controlled metabolic or immune rest might reset overactive systems. The possibility that hibernation-like states could slow inflammation and extend lifespan now moves from speculation to experiment. Nature, as always, has already performed the trials. The gut's reawakening carries emotional resonance beyond its biology. The return of appetite marks not only the end of scarcity, but the renewal of connection with the environment. Each bite, taken after hibernation, is an exchange with the world nutrients from leaves, seeds, or prey absorbed, transformed, and woven again into the fabric of the animal. Energy passes from landscape to blood to motion. The microbiome mediates this conversation, turning external matter into internal rhythm. The resumption of feeding is therefore both physical and symbolic, the body rejoining the cycle it left suspended under ice. Outside, the signs of renewal multiply. Streams run clear. Insects rise in clouds above the thawed ground. Bears leave their dens lean, but unbroken. Their first steps heavy, yet certain. Ground squirrels emerge, blinking their fur flattened by the months of stillness. Frogs thaw and call across flooded meadows. Each movement, each breath adds to the swelling chorus of spring. Inside these bodies, immune systems hum with restored balance, and gut bacteria bloom like unseen meadows dense with returning life. The precision of this reboot is astonishing. Nothing is wasted, nothing random. Every cell seems to remember its sequence, guided by chemical clocks older than ice itself. Stillness becomes preparation, not loss. Silence becomes the foundation for renewal. In the calm aftermath of winter, nature demonstrates its version of grace, a recovery so measured it appears effortless. Across the planet's temperate zones, winter does not fall equally. It drifts through valleys first, then climbs the hillsides, erasing sound and color as it passes. Forests fade into stillness. Rivers slow beneath plates of ice. And the air grows so clear that even silence seems visible. Yet beneath that quiet, the world remains in motion, rearranging itself according to the oldest schedule on Earth. Hibernation, though it seems an individual act, is woven into the fabric of ecosystems. Each creature that pauses changes the lives of many others, and the sleep of one species becomes the rhythm by which others survive. At the threshold of winter, the forest prepares, as if obeying a single conductor. Trees drop leaves to reduce water loss, roots conserve sugars deep in the soil, and the ground hardens into a shell that locks away microbes and nutrients. This transformation slows not only plants, but also the invisible decomposers that feed on their remains. Microbial metabolism drops with temperature, shifting the balance between decay and preservation. Fallen leaves persist longer, creating the layer that will nourish the next generation come spring. Even before animals retreat to their dens and burrows, the world is already holding its breath. Among the first to vanish from sight are the insects. Bees, wasps, and ants withdraw into hives and chambers where temperature stays steady. Most die, leaving behind a few queens or larvae wrapped in protective shells. Bumblebee queens burrow alone into the earth, their bodies cooled to near freezing hearts, beating less than once each second. Under the same soil beetle, larvae and solitary bees endure in silence. Their sleep determines the abundance of pollinators when flowers return, linking the fate of entire plant communities to the length of winter's pause. A season too short or too long can ripple through fields and forests, altering which blooms will set seed and which will fade unnoticed. In deeper burrows, reptiles and amphibians take refuge from cold. Snakes coil together in crevices, dozens of bodies wound into one knot of slow respiration. Frogs lie beneath mud that remains oxygen-poor, but unfrozen, their chemistry tuned to the edge of endurance. Even underground, they remain part of the surface ecosystem. Their dormancy influences prey and predator alike. Insects multiply differently when their hunters sleep. Birds migrate according to the absence of movement below. Every act of slowing becomes a signal interpreted by others, and the entire landscape rearranges itself around these intervals of rest. Above-ground predators adjust their own rhythms. Owls, foxes, and weasels change diet as small mammals vanish from view. Some follow the snowline southward. Others subsist on what remains: carcasses, roots, or stored food. Energy flow through the ecosystem narrows to a thread, a fraction of its summer abundance. Yet nothing in nature is lost entirely. Energy waits in storage, sealed in fat seeds and frozen soil. The apparent stillness of winter conceals a deep continuity. The frozen ground itself participates in this cycle. Beneath the surface, microbes switch from rapid growth to near stasis, yet their presence ensures that nutrients will be ready when thaw returns. In the Arctic, bacteria and fungi continue to operate slowly even under permafrost, digesting organic matter that has lain buried for centuries. Their metabolic whispers release trace gases, carbon dioxide, methane, and water vapor that feed the atmosphere and in turn influence climate. Each bite, taken after hibernation, is an exchange with the world. Nutrients from leaves, seeds, or prey absorbed, transformed, and woven again into the fabric of the animal. Energy passes from landscape to blood to motion. The microbiome mediates this conversation, turning external matter into internal rhythm. The resumption of feeding is therefore both physical and symbolic, the body rejoining the cycle it left suspended under ice. Outside, the signs of renewal multiply. Streams carry minerals freed by melting snow. Roots draw them upward into leaves that will later fall again. The loop closes with perfect economy, matter conserved, energy redistributed, time resumed. For scientists who study phenology, the timing of natural events, hibernation, is a cornerstone of planetary rhythm. Each year's length of sleep and moment of waking affects migration, breeding, and bloom. A shift of only a few days can break old alignments. If a warming spring draws insects from dormancy before birds arrive to feed, or if flowers open before pollinators return, ecosystems falter. The subtle coordination between sleepers and the waking world holds the balance of entire biomes. Even microscopic life follows this rhythm: algae in ice, bacteria in soil, and spores in permafrost time, their re-emergence to match the same cycles of light and temperature that guide the largest mammals. Across millions of years, these pauses have shaped evolution itself. Species that mastered the art of waiting out winter survived when others vanished. Those that learned to match their rest to the heartbeat of the planet carried that knowledge forward in genes and instincts. The hibernation of one animal became the clock for another's migration, feeding, or reproduction. In this sense, hibernation is less an escape from cold than an agreement with it, a contract written into life's machinery. To stand in a forest at the edge of spring is to witness this contract renewing itself. Beneath the thawing leaves, beetles and fungi resume the slow work of turning decay into soil. In the canopy, birds return to branches that only weeks ago seemed lifeless. Streams that lay silent under ice now carry silt seeds and the faint scent of oxygen. Every motion traces back to a winter of sleep. Energy that had been locked in bodies and seeds flows outward again, fueling the next cycle of growth. The planet's rotation dictates the rhythm. Gravity and light provide the measure. Yet hibernation refines it into music. Each participant animal, plant, microbe enters and exits at its appointed time, guided not by command but by the internal alignment of chemistry and sun. This synchronization ensures that energy never truly halts. It only slows until conditions favor motion again. The world's long sleep, far from being absence, is an act of balance that keeps life sustainable. The harmony of ecosystems relies on these rests, much like a symphony depends on silence between notes. The winter stillness allows decomposition to pause, nutrients to accumulate, and food webs to reset. Without it, the planet would race unchecked through energy, leaving exhaustion where abundance once was. The quiet intervals of hibernation grant the biosphere time to breathe. Night over the desert observatory is as still as the beginning of time. The horizon glows faintly where city lights end, and above that line the sky deepens into the color of ink. Through the telescope's lens, stars burn with a steadiness that belies their distance. To those who watch them, the question arises again: if life can sleep through winter and awaken unchanged, could it one day sleep across the gulf between planets? Scientists call the idea induced torpor. The term is clinical, but its roots reach into the same natural wisdom found in the dens and burrows of Earth. In principle, lowering metabolism to a fraction of normal could allow a human to survive long journeys where resources are scarce and time itself stretches beyond endurance. The physics are sound. The biology remains an intricate challenge. In hospitals today, medicine already uses fragments of this principle. During certain heart or brain surgeries, patients are cooled until their body temperature falls several degrees. The slowed metabolism buys surgeons time, reducing the tissue's need for oxygen and delaying injury. After cardiac arrest, controlled hypothermia helps protect the brain while blood flow is restored. Each application echoes the hibernator's logic: conserve energy, minimize damage, wait until conditions improve. Yet these intervals last hours, not months, and the return to full warmth requires precision. Even a slight misstep can cause shock or cell stress. The vision of suspended animation for space travel extends that window to an almost unimaginable scale. A journey to Mars at present speeds might take nearly a year. A voyage beyond could stretch over decades. Carrying awake active humans across such distances would demand vast amounts of food, air, and psychological endurance. If metabolism could be slowed safely, a crew could sleep through the dark interval, their vital signs barely flickering, their bodies maintaining themselves at the quiet edge of life. The challenge lies in control. Nature achieves torpor through pathways evolved over millennia: hormonal cues, temperature drops, and changes in blood chemistry that adjust each system in balance. To reproduce this in humans, researchers must understand every switch and feedback loop. Experiments on animals, rodents, primates, even pigs have shown that deep hypothermia can be induced and reversed with minimal harm, but maintaining it long-term remains elusive. The human brain's constant demand for energy resists such suspension. Still, progress comes in increments: chemical compounds that mimic the effects of cold cooling methods that protect tissues evenly, and artificial support systems that circulate oxygen and nutrients at reduced rates. The philosophical implications run alongside the biological. If a person could sleep for years and awaken without memory of the passing time, what would that sleep mean? Hibernation blurs the boundary between continuity and absence. The wood frog, the bear, the turtle all resume their lives as if time had skipped a frame. Their consciousness, however, limited, remains intact because it never truly ceased. In humans where awareness is more complex, the pause would carry deeper questions. Does identity persist when perception halts? Would awakening after decades of silence feel like survival or rebirth? Yet beyond such questions lies practical wonder. In cold storage facilities, scientists already preserve human cells, embryos, and even small organs at temperatures near freezing. The goal is longevity, without decay, a modern reflection of what evolution perfected in frozen ponds and dark dens. Each advance brings us closer to the possibility of extending that preservation to entire organisms. Cryobiology, the study of life at low temperature, now borrows its metaphors directly from the hibernators of Earth. The frog's antifreeze sugars, the squirrel's stable proteins, the bear's recycling of nitrogen, all become clues for the engineers of tomorrow's spacecraft. In the quiet corridors of space agencies, design sketches show astronauts lying in capsules, bathed in pale light, vital signs monitored by machines that never sleep. Around them, sensors would adjust oxygen, humidity, and heat with microscopic precision, mimicking the caves, dens, and mud chambers of terrestrial hibernators. Every few weeks, a pulse of warmth could awaken the sleepers briefly, allowing systems checks before the next descent into metabolic silence. The spacecraft itself would become an artificial winter-engineered stillness, guided by the biology of waiting. The connection between these visions and the natural world is more than metaphor. Understanding hibernation reshapes our concept of life's boundaries. For centuries, the line between living and non-living seemed distinct: motion on one side, stillness on the other. Hibernation erases that clarity. It shows that life can slow to invisibility, yet remain intact, that stillness can contain continuity. If those principles can extend beyond Earth, they may define how life endures in environments where no immediate energy source exists. Space is the ultimate winter cold, airless and nearly infinite. Any organism that learns to rest through it would not only survive, but might thrive when awakened. The first to test such endurance will likely be machines carrying traces of biology, cultures of cells, tissues engineered to respond to sleep and reawakening. If those can cross space unharmed, the next step will be human. And when that day comes, the journey will mirror the rhythms written into Earth's smallest sleepers. As we imagine these futures, it becomes clear that the knowledge we seek already resides in the planet's oldest teachers. The frog, frozen solid in a forest puddle, the turtle beneath ice, the bear in its den, all hold answers to questions we have yet to ask. They show that survival is not always speed or resistance. Sometimes it is restraint, the wisdom to do nothing until time itself changes shape. Outside, the observatory dawn approaches. The telescope cools in the fading night air. To the east, the horizon brightens, the same light that has woken every hibernator since life began. The researcher closes the notebook, knowing that each discovery made here will circle back to the same truth: life's capacity to pause is as extraordinary as its capacity to move. As the season turns fully toward warmth, the last traces of snow disappear into the ground, carrying the memory of ice into roots and rivers. Evening falls softer now, the light lingering a little longer each day. Somewhere in the distance, the first night insects sing their wings, drawing lines through air newly thick with scent. The world feels lighter, yet its stillness has not gone. It has simply become part of the motion folded into the pulse of living things. Perhaps this is the real lesson, that every pause contains the seed of renewal, and every motion owes its grace to the rest that came before it. In that understanding lies a model for balance, one written not in words, but in breath temperature, and the steady patience of life waiting for its moment to return. Good evening, and thank you for drifting through this journey tonight. May your dreams rest easy, and may your thoughts drift among the stars.